Imagine for a moment your body needing to quickly build a critical recognition tag for a cell, or maybe deploy a rapid, powerful defense against some kind of molecular attack. Where do these specialized tools and the raw materials come from? It's way more intricate than just burning glucose for immediate energy. It's really about strategic building protection, communication. So let's dive deep. Today, we're zooming out a bit from our usual focus on glycolysis and glycogen. We're doing a deep dive into Chapter 27 of Marks Basic Medical Biochemistry, our mission, to basically uncover these hidden but absolutely vital pathways that use glucose for more than just fuel. We're talking the pentose phosphate pathway. Think of it as the cell's unsung hero for building and protecting. And then we'll get into the surprisingly complex world of how our bodies construct and use these intricate sugar structures, things like glycosides, lactose, and those really important glycoproteins and glycolipids. The goal is to quickly arm you with the essential understanding, connect it to real-world medical stuff, and give you those aha moments. All without needing the textbook right in front of you.
Yeah, what's truly fascinating here is how glucose, our fundamental energy source, it's just incredibly versatile. It's not just about fueling your sprint, right? It's about providing the absolute bedrock for DNA, building the sophisticated identity cards on your cells, and arming your body against oxidative stress. This chapter really illustrates that hidden depth, the sheer ingenuity of, well, cellular chemistry.
Okay, so we're starting with what I sometimes think of as the cell's strategic supply depot and defense hub, the pentose phosphate pathway, also known as the HMP shunt. So instead of just burning glucose for ATP, this pathway takes glucose 6-phosphate and turns it into two incredibly important products, each with distinct life-sustaining roles.
That's right. Think of it as having two main jobs running in two phases. You got the oxidative phase that's all about defense and building, and then the non-oxidative phase, which is more about flexibility, making adjustments as needed. Every single cell in your body needs what this pathway produces.
All right, let's peel back the layers on that first part, then the oxidative phase. Glucose 6-phosphate goes through a couple of clever chemical changes here. The main result is creating ribulose 5-phosphate, a molecule of CO₂, and maybe most importantly, two molecules of NADPH. Reduced nicotinamide adenine dinucleotide phosphate, that's a mouthful, but it's a game changer, isn't it? What exactly is this NADPH, and why is the reduced part so critical?
Well, NADPH is essentially the cell's specialized electron carrier. It's crucial for building new molecules biosynthesis and for neutralizing harmful ones. When we say reduced, it just means it's carrying high-energy electrons ready to donate them. And connecting this to the bigger picture, NADPH is really the cell's main reducing agent. It acts like an internal antioxidant and a master builder, all rolled into one. See, the cell keeps a very high ratio of NADPH compared to its oxidized form, NADP plus NAD, so it's always ready, basically, to donate those electrons for these vital reductive reactions.
And these reactions are happening constantly. We're talking about critical processes for making things like fatty acids and cholesterol fundamental building blocks. But maybe even more dramatically, NADPH is absolutely vital for detoxification, like helping your body process certain drugs, and for defending cells against reactive oxygen species, those damaging free radicals that can, wreak havoc. It directly powers the glutathione defense system. That's like a cellular shield, right? Essential for every cell, but especially red blood cells.
Precisely. And the other key product from this oxidative phase, that ribulose 5-phosphate, it quickly gets converted into ribose 5-phosphate. And this pentose sugar, this 5-carbon sugar, is the indispensable backbone for making nucleotides, the fundamental building blocks of your DNA and RNA. So whether the cell needs to build new genetic material or beef up its defenses, this oxidative phase of the pentose phosphate pathway is absolutely essential.
Okay, but what if the cell's needs change? What if it needs, say, more building blocks for DNA than it needs antioxidant protection right now? That's where the incredible flexibility of the pathway's second stage comes in, the non-oxidative phase. This part is all about shuffling sugars around, isn't it?
Yeah, and this raises an important question.
Yeah.
How does the cell actually balance these demands? Well, this non-oxidative phase is completely reversible. It lets ribulose 5-phosphate be rearranged into other sugars. You can make more ribose 5-phosphate if you need it, or you can even convert things into intermediates that feed right back into glycolysis, like fructose 6-phosphate. It's this dynamic interplay, making sure the cell always has what it needs when it needs it.
And a key player in this sugar shuffling act is an enzyme called transketolase. It's got a pretty cool clinical connection too, right? It uses thiamine pyrophosphate that's related to vitamin B1. Thiamine is a coenzyme, which means for you listening, doctors can actually measure the activity of this enzyme in red blood cells with and without adding extra thiamine to diagnose a thiamine deficiency.
That's a perfect example, exactly. How these specific enzyme functions have direct diagnostic relevance.
Eh.
We actually talked about this with a patient, LM, in a previous chapter discussion. He was diagnosed with beriberi heart disease, which is a direct result of thiamine deficiency.
Right. And Alam's story, unfortunately, continues to highlight another critical clinical insight connected to this pathway: glucose 6-phosphate dehydrogenase deficiency, G6PD deficiency. This is a defect in that very first really critical enzyme of the pentose phosphate pathway, the one that makes the NADPH.
Yeah, and for red blood cells, which rely almost exclusively on G6PD to make their NADPH, this is a huge problem, a massive vulnerability.
So without enough NADPH.
Without enough NADPH, red blood cells just cannot maintain their crucial levels of reduced glutathione. They can't protect themselves. This makes them incredibly vulnerable to oxidative stress. And that vulnerability can trigger what's called acute hemolytic anemia. Basically, red blood cells get destroyed rapidly.
If the person is exposed to certain triggers.
Exactly. And those triggers can be surprisingly common things. Certain drugs, like sulfa drugs or some antimalarials, specific infections can do it. Or even famously eating fava beans.
Wow, so just eating fava beans could trigger a crisis.
For someone with G6PD deficiency, yes, a casual lunch could turn into a cellular crisis.
Oh.
LM, again, is a stark reminder. He got a fever from an infection, was given trimethoprim sulfamethoxazole, that's a sulfa drug combo, and within days, boom, jaundice, his hemoglobin plummeted, his urine turned red-brown from all the free hemoglobin spilling out. It pointed straight to acute hemolysis, a really tragic interplay between his underlying G6PD deficiency, the infection, and the drug he was given.
That's quite something. And it's common, you said.
It's actually the most common known enzymopathy globally. Affects something like 7% of the world's population. Many people live completely fine, asymptomatic, until they hit one of these oxidant challenges. Interestingly, its geographic distribution overlaps a lot with areas where malaria is or was prevalent, which suggests it might actually offer some protection against malaria, a kind of evolutionary trade-off, perhaps. And biochemically, there's a need regulatory loop. NADPH itself strongly inhibits the G6PD enzyme. It's a feedback mechanism ensuring supply meets demand. Clever, really.
So, okay, we've seen glucose as this unsung hero, constantly fortifying our cells, cleaning up damage, but its versatility goes even further, right? It becomes the architect of our cellular identity. Let's shift gears now and explore how it builds those more intricate, larger sugar structures that define us, literally from our blood type to how our cells recognize each other.
Indeed. The cell's ability to interconvert sugars and build these complex sugar derivatives often relies on what we call activated sugars. These are basically sugars that have been chemically tagged, usually by attaching them to a nucleotide, things like UDP-glucose or UDP-galactose. This activation provides the energy needed for building these complex structures.
Right, so think of these nucleotide sugars like the cell's perfectly packaged delivery trucks for sugars. They make sure the right sugar gets to the right place with enough energy to forge a strong bond. And UDP-glucose, in particular, sounds like the Swiss Army knife of sugar precursors. It seems incredibly versatile, ready to build almost anything from energy stores to these cellular identity tags.
It truly is. UDP-glucose is a precursor for so many vital compounds. Glycogen for energy storage, lactose, which we'll get to, UDP-glucuronate, crucial for detox. And it's absolutely essential for building the complex carbohydrate chains found in proteoglycans, glycoproteins, and glycolipids. The sugar gets precisely transferred from the nucleotide sugar to form a new glycosidic bond, basically. A strong link between sugars and the UDP part acts as an excellent energy-releasing leaving group.
Okay, one of UDP-glucose's most impressive transformations seems to be into UDP-glucuronate. That happens by oxidizing UDP-glucose at a specific carbon atom.
That's it. And once created, UDP-glucuronate becomes one of the body's primary detoxifiers,
Yeah.
especially for hydrophobic compounds, things that don't mix well with water.
Ah, detoxifiers. How does that work?
Well, this highlights how vital even small chemical tweaks can be. Glucuronate residues, they carry negative charges. They get precisely added to these hydrophobic compounds. This dramatically increases their water solubility, transforms them really, makes them much easier for your body to excrete, usually in urine or bile, prevents them from building up and causing toxicity. And this applies to a huge range of things: drugs, environmental toxins, even some of the body's own metabolic byproducts.
A classic example and a very visual one is bilirubin, right? That breakdown product from heme and red blood cells.
Exactly. On its own, bilirubin isn't very soluble. But in the liver, glucuronate residues from UDP-glucuronate are skillfully attached. This creates conjugated bilirubin, which is now soluble enough to be excreted into bile.
And if that process falters, that's when we see jaundice, the yellowish skin and eyes.
That's often caused by the accumulation of the unconjugated insoluble bilirubin. It's actually a common sight in newborns called neonatal jaundice. It happens because they often have increased red blood cell destruction right after birth, plus their liver's bilirubin conjugating system isn't fully mature yet.
Right, and for those newborns, there's that fascinating treatment,
Yeah.
phototherapy,
Yeah.
putting them under special lights.
Yeah, it's quite neat. The light causes chemical changes in the bilirubin itself, making it more water-soluble directly, so it can be excreted more easily, essentially bypassing the need for the still-developing liver conjugation system. A clever biochemical workaround.
Okay, another fascinating use for a derivative of UDP-glucose, specifically UDP-galactose, is in making lactose, milk sugar. And here's a cool fact. Your body doesn't actually need dietary galactose to make lactose.
That's right. Galactose can be synthesized directly from glucose inside your body. It involves an epimerization reaction, flipping a hydroxyl group on UDP-glucose to make UDP-galactose. And lactose itself, the sugar in milk, is synthesized exclusively in the mammary gland during lactation. The enzyme responsible is lactose synthase. It actually has two subunits, a base enzyme called galactosyltransferase, and this remarkable modifier protein called α-lactalbumin.
Okay, and here's where it gets really interesting as I understand it. During lactation, this α-lactalbumin protein kind of flicks a switch on the galactosyltransferase, right? It dramatically boosts its ability to make lactose specifically.
Exactly. It lowers the enzyme's requirement for galactose and increases its affinity for glucose. So it basically shifts the enzyme's focus, turning it into a dedicated lactose factory after childbirth. Before lactation, that same galactosyltransferase might be busy building other complex sugars on glycoproteins.
So that answers a common question then. A lactose-intolerant pregnant woman can still breastfeed successfully?
Yes, absolutely, because her body is manufacturing the necessary galactose component from glucose, not relying on dietary lactose. It's a brilliant biological adaptation.
But things can go wrong. What about conditions like galactosemia? What happens when there's a glitch in handling galactose itself?
Right. This shows how these metabolic blocks can have these cascading effects. In classic galactosemia, you get a buildup of galactose 1-phosphate, usually because an enzyme needed to process it is deficient. This buildup basically gums up the works for glucose metabolism too. It inhibits phosphoglucometase, which is needed to get glucose out of glycogen stores, so you get hypoglycemia, low blood sugar, and it also seems to interfere with making UDP-glucuronate, which reduces bilirubin conjugation, leading to jaundice. So one single enzyme defect causes multiple serious clinical problems.
Wow. Okay, so we've seen these nucleotide sugars as crucial building blocks for, let's say, simpler complex sugars like lactose and glucuronides. Now, let's talk about how they become the architects for the really grand structures, glycoproteins and glycolipids. These sound incredibly important for cell function, recognition, the very structure of cells.
Absolutely. Glycoproteins are proteins with carbohydrate chains attached. Glycolipids are lipids with carbohydrate chains attached. They're integral to cell communication and identity. They form recognition sites on the cell surface for, well, everything. Hormones, other cells, even viruses latch on to specific ones.
Let's tackle glycoproteins first. These have short, often branched sugar chains, oligosaccharides attached to proteins, and they seem to be everywhere.
There really are. Hormones, antibodies defending your body, enzymes involved in blood clotting, structural bits like collagen, even the protective layer of mucus. Many are secreted out of the cell, others are destined for specific organelles like lysosomes, and many are embedded in cell membranes acting as receptors, transporters, or helping cells attach to each other.
And the synthesis sounds complex, like an orchestrated dance.
It is quite orchestrated. The protein part is made first, usually in the endoplasmic reticulum, the ER. Then these intricate carbohydrate chains are added and modified as the protein moves through the ER and then the Golgi apparatus. For some types, like N-linked glycoproteins, the core sugar chain is actually built first on a special ligand anchor called dolichal phosphate, and then the whole thing is transferred onto the protein. And crucially, as we'll see, some of these carbohydrate chains act as specific address labels or markers, directing enzymes to their correct destinations within the cell, like the lysosomes.
Okay, and this brings us to a really devastating clinical insight. Eye cell disease. This is a rare, severe condition where the cell's internal postal service basically breaks down. Lysosomal enzymes lack that crucial address label.
Yes, and this is where we see just how incredibly precise these biochemical processes need to be. The defect is in an enzyme called a phosphotransferase. located in the Golgi. This enzyme's job is to add a specific tag, a mannose phosphate group, onto enzymes that are destined for the lysosome. This tag essentially says, deliver to lysosome. Without this tag, these vital digestive enzymes are mistakenly packaged up and secreted out of the cell instead of being delivered to the lysosomes, the cell's recycling centers.
And the consequence is severe.
Very severe. The lysosomes within the cells are starved of their normal enzymes. So waste products, molecules that should be broken down, just accumulate inside the lysosomes. These form characteristic inclusion bodies, which you can see under a microscope. They literally clog up the cells, leading to progressive cellular dysfunction throughout the body. It affects multiple organ systems.
Wow, okay, shifting to glycolipids now. These are derivatives of a lipid called sphingosine, right? Things like cerebrosides and gangliosides. They have a ceramide base with various carbohydrate structures attached, and their job is mainly intercellular communication and cell recognition.
That's right. Their synthesis also happens in the Golgi. The lipid part anchors them firmly into the cell membrane, while the carbohydrate portion extends outwards into the extracellular space, acting like an antenna or a recognition signal. There are critical signals. For instance, the toxin produced by cholera bacteria specifically binds to a glycolipid called GM1 ganglioside to gain entry into intestinal cells.
So what's a really classic example of these glycolipids in action that affects pretty much all of us?
That would have to be the blood group antigens, the ABO system and the RH system. These are actually oligosaccharide components, the sugar parts, of glycolipids and some glycoproteins found on the surface of your red blood cells. And they are what determine your blood type. Our genes dictate which specific enzymes, glycosyltransferases, we have. These enzymes are responsible for building these carbohydrate chains. Most people start with a base structure called the H substance. If you have the gene for the A transferase, you add an N-acetylgalactosamine sugar that makes you type A. If you have the B transferase, you add a galactose sugar that makes you type B. If you have both enzymes, you have both sugars making you type AB. And if you have non-functional versions of both transferases, you just have each substance, and that makes you type O.
And this has huge implications for blood transfusions, of course. As Edna R., who we mentioned works in a hospital blood bank, knows very well, people naturally develop antibodies against the A or B antigens they don't have on their own cells, right?
Exactly. So someone with type AB blood has both A and B antigens on their cells, so they don't make antibodies against either A or B. That's why they're called universal recipients for red blood cells. Conversely, someone with type O blood has neither A nor B antigen on their cells, but their plasma contains antibodies against both A and B. This makes their red blood cells safe for potentially anyone, the universal donors, but they can only receive type O blood themselves.
So going back to Edna's patient with type AB blood, they could safely receive type A red blood cells.
Yes, because the patient's own cells already have the A antigen, so they don't have anti-A antibodies that would attack the donated type A cells.
But receiving type A serum or plasma would be a problem.
That would be a big problem because type A serum contains anti-B antibodies, and the type AB patient does have B antigens on their cells. Those antibodies would attack the patient's own red cells. It's a critical distinction in transfusion medicine, highlighting just how specific these molecular recognition events are. And of course, the Rh sister, particularly the D antigen, is another major factor, being highly immunogenic and critical for safe transfusions.
Okay, finally, let's touch on the sphingolipidoses. These sound like a class of really devastating lysosomal storage diseases. They arise from defects in breaking down glycosphingolipids.
Yes, exactly. It's a problem with degradation, not synthesis. This is tragically illustrated by cases like JS, a toddler showing severe developmental delay and muscle weakness, ultimately linked to Tay-Sachs disease. These diseases happen when specific lysosomal enzymes, the ones needed to break down these complex lipids step by step, are deficient or just don't work properly. As a result, the specific lipid that enzyme was supposed to break down accumulates within the lysosomes. This causes progressive damage and degeneration of affected tissues, especially the brain, but also skin, and the reticuloendothelial system cells.
And Tay-Sachs disease itself, it's an autosomal recessive disorder, right? More common in certain populations.
Yes, it's particularly prevalent among individuals of Ashkenazi Jewish descent. The core defect is in the alpha subunit of an enzyme called hexosaminidase A. Often it's due to a specific gene mutation, like a small insertion that messes up the protein structure and makes the enzyme non-functional.
So with hexosaminidase A not working.
With hex, a non-functional, a specific glycolipid called GM2 ganglioside cannot be broken down. It accumulates massively, primarily within neurons. This relentless accumulation causes the characteristic severe psychomotor deficits, blindness, seizures, and ultimately fatal neural degeneration seen in Tay-Sachs. And there are related conditions too, like Sandhoff disease, where a different subunit, the beta subunit, is affected, knocking out both hex A and another related enzyme, hex B, or Sandhoff activator disease, where the enzymes are fine, but a crucial helper protein is defective. It just underscores how precise and delicate these biochemical pathways are.
Okay, let's try and bring this all back home then. This deep dive into these really intricate biochemical pathways has shown us a lot. The pentose phosphate pathway isn't just some metabolic side road. It's your cell's critical internal arsenal and construction yard. It's constantly churning out NADPH, the vital tool for building things and defending against oxidative stress. And it gives us ribose 5-phosphate, essential for making our very DNA and RNA.
Right, and beyond that, we saw how these nucleotide sugars, like UDP-glucose, are the activated building blocks for all sorts of complex carbohydrates. Glucuronides emerged as your body's essential detoxifiers, making nasty compounds water-soluble so you can get rid of them. And glycoproteins and glycolipids are just vital for everything, defining your blood type, cell recognition, cell structure, communication. The list goes on.
We've also seen the really profound, often devastating clinical consequences when these pathways go wrong. From G6PD deficiency, causing red blood cells to break down after eating fava beans, to eye cell disease, where lysosomal enzymes get lost in the mail, leading to widespread cellular chaos, and the sphingolipidoses like Tay-Sachs, where a single enzyme defect leads to catastrophic neurodegeneration. So the next time you think about glucose, remember it's not just fuel, it's not just quick energy, it's also the fundamental building block for these incredibly intricate systems that protect your cells, detoxify your body, define your blood type, and even govern how your cells talk to each other. It's a truly brilliant molecule.
Absolutely. And considering how interconnected and finely tuned all these pathways are, makes you wonder, doesn't it? What other unexpected clinical challenges might pop up from a seemingly small glitch somewhere else? A tiny error in a sugar modification, or maybe another enzyme's activity. The world of biochemistry is truly this complex, vital web of connections, always waiting for your next deep dive.